Do you hear that voice, that lady?
Yes.
And she's in French?
No.
No. It's not French for me.
Okay.
I mean, I was going to make a snide remark about the invite being in French where I'm like, I don't know. How do we want to get in?
We'll have to edit this part out, right?
No, it's hilarious.
Okay.
Hey, hello everybody. So welcome to this very second podcast of the ChUG group in Chicago. ChUG is dedicated to all things cytometry. My name is David Leclerc. I'm the technical director of the CAD facility at the University of Chicago.
I am Ryan Duggan. I'm a senior scientist at AbbVie, just north of Chicago.
We're happy to host Kelly Lundsten today.
Yeah, I'm Kelly Lundsten, currently doing independent consulting with a specialty in fluorescence chemistry and its applications.
And we were just discussing before starting recording, Kelly managed to score the cytometry.me email,
Yes.
web address for a website, which is pretty awesome and well deserved. So
Although I'm not really sure what .me means.
I don't know.
.me.
Hmm.
Meh, whatever.
It's probably some random island off the coast of the Atlantic or something like that.
Yeah.
So today we wanted to record a podcast about fluorophores, and the reason I thought this was interesting is that over the last couple of years we've had a bunch of new dyes coming our way, and they are all named very strange things. We had the BV dyes, BUV, BB. There's the super bright dyes from Thermo Fisher, and now Cytec is throwing their own dyes, and BioLegend has the Fire Dyes series. Sony came up with Curavia, and Phytonex with Nova Fluors, which are tunable dyes now. And so, I don't know, when I started working here, we had a bunch of dyes that were essentially all the same. So you had the PAC Blue, E-Fluor 405, and then, the Bio Blue, and they were all essentially shining in the same detector, so we could kind of use them without thinking about it. But nowadays, it seems like all of these guys are slightly different, and you need to learn a little bit more about what's going on with these dyes. And so that's why Kelly is here. She's going to teach us all that stuff.
Yep. I was going to say, most of those that you said, they literally are the same thing. They're just named different things by different companies or for slightly different reasons.
So do you want to talk about, like, start with the chemistry of the dyes, or just like why are things being thrown our way as it is?
Yeah, I always say like spectra is like a fingerprint for the dye. If your spectra of Alexa 405 and Pacific Blue, and if they literally overlap and they're within like a nanometer or so variance, they're basically the same chemical structure. Maybe they added a sulfonate, maybe they added a chlorine, maybe XYZ, but it's mostly people wanting to market and name things. So how do we make the slide presentation?
You can actually share your screen.
Oh,
I know.
see, you guys are on these things all the time, but now that I'm at home, a consultant, it's not, I don't do this.
Well, while you're getting that set up, also, don't you think like the evolution or the launch of all these different dyes is pretty much directly correlated to spectral cytometry? How much of it is because of that?
I think that it's kind of two-pronged. First was that we really needed to just have better alternatives to like protein tandems, like PE size 7 and APC size 7. Like there were better options than just, so some of it is just kind of making things that were old just a little better because the acceptor fluors were getting a little better. So there's that, just rehabbing the old. And then absolutely anything that's like a brand new spectra, things that we hadn't thought about before. You know, one of the things that had been really frustrating when we were launching the Brilliant Violets had been that the Q dots had already determined what our acceptors would be. You know, with spectral, if that's how we had started, it would have looked entirely different spectrally with how everything is placed than it does. But, you know, we start with, you know, the 3 color, the 4 color, to the 8 color, to the 12 color, to the 18 color. and then we're always building on top of those things. And so we're trying to fit spectra into the holes off of each of those lasers. So yeah, I would say spectra is definitely, spectral is very much the reason why we have a lot of the new tandems. But I would say that the pressure on the market was because of the brilliant dyes and how they were polymerized. And I am gonna talk about that in this, like one of the biggest limitations of simple organic chemistry is simply, you only have one optical unit. You have one conductive, semiconductor basically. And because of that, you're always gonna be limited. You can only emit as much energy as you absorb. So, and then you can't emit more than it. It's always gonna be an attrition from that point. So unless you can get many of them onto a single biomolecule, like a polymer, that's the only way to get them brighter. So I think there were a lot of pressure for single molecule that the brilliant violets had let us know we could have, something that was more stable than PE. So that's what everybody's looking for: brighter, more stable to solvents, to light, more spectrally tunable, easier to conjugate and mix and match. There are all these things that, and limitations to previous technology that kind of over time, we realized we could overcome at each stage, right?
Mm-hmm.
And so that's basically in total what you're calling better, right? So that yes. It's better because X, Y, and Z, what you just said.
That's what I say. Not all dyes are better. Some dyes just are new. Especially when we're talking about like the fire dyes that spark dyes that you were talking about before. They're still just simple organic chemistry. They're still going to be really limited in brightness and in photostability and all those things, but they still give us another floor for a spectral. So does that make them better? No, it doesn't make them better. They still have a ton of limitations, but it makes another option. Versus the brilliant violets were significantly better than Q dots. Did you guys ever use Q dots?
We did use Q dots back in the day. Yeah.
Yeah.
Ugh. I think what are they used for now? TVs or something?
No,
my TV uses a Q dots technology.
Q dots have always been really popular for microscopy.
No.
Mostly because of the, you really have to zap them with a laser. You have to ablate them in order for them to lose fluorescence. So for microscopy and single molecule tracking, that's like, it's a pretty big deal.
So at one point, BD was using a 325 HECAD on their LSR2, which lasted roughly 6 months before it started dying. Yep. But the Q dots actually looked really good on there.
Yep.
So.
Yeah, the biggest issue, which we can talk about when we get to that section, with the Q dots from the molecular probes perspective, had been conjugation. When Q dots first came around, most of us weren't past 12 colors. So you couldn't just make a whole bunch of antibodies and be like, oh, we know that these ones are going to be relevant to researchers. And they degrade the antibody faster because they're conjugated to the hinge region. And that kind of conjugation in general is never going to increase the affinity of an antibody, and especially at the hinge with a bowling ball. So that was one of the hardest things, was how do you get people to adapt to something like a bowling ball conjugated to your antibody? If they have to conjugate it themselves, and the biggest issue there was the purification, not even the conjugation itself. It was how Q dot's humongous. And all you have is this Q dot with a whole bunch of antibodies conjugated to it. How do you purify those two things apart from each other? The molecular weight difference isn't that significant. So it was just really hard to get adoption for those, let alone they just kept failing QC like every other day,
So just to close.
Conjugating them to like CD4 wasn't going to help anybody. Like the last thing you needed was Q dot 655 or 650 or whatever it's called on CD4. It's going to screw up a lot of things in your assay. So,
right. So just to close that loop, would you say that, and back to Ryan's question, like the Aurora is bringing up the possibility of new dyes being used, was a limiting factor before spectral flow, and saying Aurora is that Sony is there too? Like,
Yeah, fluoroflow
But
cytometry.
before that was the limitation, not only the lack of detectors, but like just the bandpass filters that you'll find most commonly on most instruments. And so if you had a cool, you know, blue laser excited dye that would shine at 610, you would necessarily have the proper detector for it, so you would not develop a molecule like that.
Kind of. I mean, you're hitting on basically every single thing that got us to where we are now. You had instrumentation limitations. So lasers, efficiency, filters, whatever. And also background in that range. Why difficult to see blue all the time, right? There were also limitations in the chemistry. The brilliant violet polymers, they weren't even invented. You know, they weren't invented for this kind of optical purpose until we were into the 2000s. And same with the DNA fluors as a, you know, DNA as a backbone type fluors. That didn't come around until 2007. So it wasn't just, this is my point. It wasn't just that we needed a spectral system. We needed, you know, the spectral system couldn't even happen until we could integrate that many channels at a time. There were so many technological limitations. I feel like the chemistry would pull and be like, hey, we've got this new XYZ, and the machine would be like, I don't know, I can't see that yet. That doesn't help me. And then all of a sudden one day they would change, and then reagents would be the ones that were holding everybody back, you know, back. I find right now when I'm trying to make a panel of like more than 24 parameters, like how many markers do you really need to see? I start wondering, you know, maybe I can glance over top of a million, you know, like 40 some different markers, but right now biologically we often don't really need to get that high for most purposes. And then you start to transition into single cell sequencing. So I'm just trying to say that there's a need, or there's not. There's the technology, there's not a technology. There's chemistry, there's not chemistry, but each of those is a push and pull on the thing that's next to it. So yes, spectral allowed us to see all these things, but that's not the only thing that allowed us to get into polymerizing DNA as a backbone. That's a whole, you know, thing that came before for many, many, many years.
Yeah. All right. You want to dig into chemistry of everything?
Yeah, but I shared my screen. Seriously, David. Oh. David, you're muted.
On my end.
That looks good.
That looks great.
So we are good to go.
Okay. Okay. Synthetic organic fluors are probably the oldest type of fluorophore that we've used, right? Like I think fluorescein is probably the first. They come in kind of discrete family names. Like none of this is marketing. These are actually, you know, the limited family, the chemical family groups from which we can derive synthetic organic fluorophores. Coumarin is always going to be the smallest, and blue inherently as a single conductive unit just can never be bright. It can't absorb enough energy. It doesn't have a high extinction coefficient. And so it just can't be bright. And on top of that, all of our biology is autofluorescent in blue. So that was always, you know, when I was back at Molecular Probes, I thought that that was a problem that we would just never be able to overcome, because how can you, besides making them into a polymer, you can't make them be something they can't be structurally. But the strength is, you know, strength as in the only thing that they're valuable for is that only this sort of structure, only this tiny structure is going to be sensitive to high frequency wavelengths, like the short wavelengths, violet and UV. So just inherent limitations to conducting energy. The second and most popular, that most everything that we use is rhodamine, which is this xanthine ring, is that top ring, those three hydrocarbons up top. Those we can derive into a bunch of different things, but we can't make them blue and we can't make them much past 650 emission. You're never going to get into the near infrared with a xanthine because it becomes too hydrophobic, but still makes up Texas red. It makes up Alexa 610. fluorescein, Alexa 568, all of those are rhodamine derived fluors. So helps us for 1,000,002 things. The cyanine dyes, entirely off patent. Actually, both the rhodamine and cyanine dyes are all off patent now. So all the Alexa fluors are finally off patent. So that's another thing I think you're going to see in the industry now. Why would any of us pay royalties to use the name Alexa fluor if we don't have to? Because that's the only thing that you would be licensing at this point is the term Alexa fluor. Other than that, all of us can find much cheaper sources for those same exact sulfonated structures that make up the Alexa fluors. So I think you're going to find everybody doing that. And that's going to make it also really confusing marketing wise, because you are just going to have one company make up all these new names for things, and you'll have no idea if they're new or different, or if they're special or better or anything like that. Cyanines tend to be really good acceptors because they are big. Anytime a molecule is big, it usually has a higher extinction coefficient because that absorbing of energy is around those cyclic rigid hydrocarbons. But that size, that bigness, that, how big they are can also make them more susceptible to reactive oxygen species, make them more specific to, make them more susceptible to non-specific binding. I'm sure you guys have you guys seen like Cy5 and Cy7 non-specifically bind on some subtypes? Yeah, I used to do that a lot. Well, because we were developing that monocyte blocker at BioLegend. So I saw it even with Texas Red and some rhodamines as well, but cyanines, it's definitely exaggerated. There's the Bodipi dyes, those are also off patent. Mostly we use those to label anything that's lipophilic, like LDL or something that's going to compartmentalize to a small hydrophobic portion of a structure. They are really nice because they're super, super small. We can make cell permeant probes out of them. Cyanine dyes can't be cell permeant. They're way too big. The only ones that can be are like CFDA, the fluorescein xanthine derived fluor. But that molecule gets, the harder it's going to be to deliver intracellularly. So that's actually why 2 of the new modifications that I think we'll start to see, these are used in microscopy already. So far, when I was testing some of these probes from their sources, siliconized rhodamine is a substitution of this, I don't know if I can, I don't think you can see my pointer, right? It would have to be David's. It's basically just a substitution of a silicon into, or silica, into one particular point in the ring structure. And what it does is just make the whole ring more structurally stable, and it rotates less, so it's losing less energy. And that helps make it a little bit brighter. And generally, and it's actually the same with the azetidine modification, which you can kind of see that structure better. The structure always tells you, I know nobody ever wants to look at structure, but the structure always tells you what is special or what that fluor is designed for, or even the wavelength. You can, the bigger the structure gets, the more near infrared it's shifting. And same with this little azetidine ring off of those nitrogens on either side. They're also holding a rotating element of the fluorophore in check. It's like putting like a cap on either side so that it remains rigid. One of the easiest ways or best ways of increasing brightness is increasing the rigidity of the fluor. Fluors are planar, and it's important that they're not losing energy through vibration or rotation at any part of the structure. So that's what they're doing. They're just trying to bind it up so that it's more rigid. The little thing on the right here is from Luke Lavis, because both of the siliconized rhodamine and the azetidine modification, Luke is at Genelia Farms. He's an amazing scientist in terms of fluorescence. And he does a lot with like live cell loading of both of these siliconized rhodamines and azetidine modified probes, and doing actually in-cell probing and in-cell detection, which you can find all over. So go to the next. Oh, wait. Go back. So generally, all of these are only ever going to be so bright because you can only conjugate so many of them to an antibody. They have, and that brightness is a direct relationship to what you perceive as photobleaching or tandem transfer of energy. You know, if you have a polymer of 10 fluorophores, or you have one antibody with maybe 3 fluorophores, as you photobleach each of those fluors away, they're gonna seem to photobleach faster to you because you're just detecting them by your machine. Versus if you have 10, as you lose 1 or 2, you're not gonna notice it quite as much because the ratio of fluorescence is still there. You're still a sustainable signal to do imaging. Where we notice them constantly, and especially in spectral, is that tandem transfer of energy, where we see our spectra shift. We have entirely different ratios in 2 different channels, and it's going to be harder to accurately unmix those signals from each other. So we can't do without these. They're still going to be the acceptors for, even when we go into DNA backbones or into the BV-like backbones, we're still incorporating these same type of fluorophores into those backbones as we polymerize them. We can't ever get away from this. This is what color is, literally. All of these are colorful as well as fluorescent.
So any dyes will be based on that structure, and then you kind of.
Mm-hmm. Yeah, green is always still going to be green with this structure. You can't manifest green some other way, unless you talk about nanocrystals. That's different. But.
Right.
All right, so protein fluors. Everybody is going to be familiar with GFP. Well, you know, the reason we like those is because we can mutate them into the entire fruit repertoire, because, you know, just through different combinatorial relations in the structure. The thing that most people don't realize is when they think of GFP as being bright, same with RFP, they're not monomers, right? They're dimers and trimers, or, you know, that protein is finding a partner and pulling, each of those GFPs is finding a partner. And then they're becoming brighter as a single molecule in your mind because you just had an additive ×2 effect. So finding a bright fluorescent protein is exactly the same problem for them in these applications as it is for us with simple organic chemistry. You know, it's great if we can polymerize them, they're super bright. But, so biggest issue with these guys is obviously as they're translated on the protein that's being endogenously expressed, you can have cytotoxicity or conformational changes or changes in affinity. But I would say for us, the biggest issue is going to be just stability. Oh my gosh. I mean, like the 1,000,002 times in tech services that you're, does your, you know, PFA contain methanol? Did you shortcut it by not just letting it spin for 12 hours on the spinning plate? Instead, you add some methanol to bring it into solution to disperse it. You know, like when I was in neuroscience, we prepped our PFA fresh every single time because morphologically we would see a difference in our neurons because neurons are so sensitive to fixation. So people that are buying commercial fixatives, not so much problem, or they just make one monster batch and just keep pulling from the same batch. Maybe they won't see as much of a problem either. But so phycobility proteins, there's actually still some fun stuff going on here. I don't know if you know of Columbia Biosciences. They have a whole bunch of really interesting, oh gosh, and actually this link that I have here, it lists all these different sources of PE, APC, phycocyanine, and all the different bacteria that, you know, and where that bacteria is. You know, theoretically, one company could be sourcing from an entirely different bacteria their PE versus somebody else, right?
Oh, right.
At Probes, we used to have this like I don't know if this actually was true, but they said that we stayed in business once because there had been a tsunami or something had happened off the coast of Japan, and it had destroyed all of the source of PE for like one supplier. And so the joke was that Molecular Probes stayed in business when they were just open for like 10 years because they became like the sole suppliers for PE for the world. I don't know if it's true, but it was just kind of like a little funny story. But the so I mean, PE and phycobility proteins, they are are nature's natural polymerizer or polymerized protein. None of those curls there, none of that protein is fluorescent. The fluorophores are embedded in that like it's a matrix, like it's a scaffolding. So actually the second down below, that is like one leaflet of PE, and the white little structure in there, that's the actual phycobilin, and the phycobilin itself is the fluorophore. So this is just nature's way of creating this big complex protein mesh that holds and isolates the chromophore, holds it tight, because otherwise it's, again, it has to be rigid. So it holds it tight so that it can be fluorescent. And again, if we apply any sort of solvent to it, like phosphoprotein detection and stuff like that, you denature that protein, it relaxes the fluor, and you lose fluorescence. So if we could make, like probes once tried to make a fluorescent dextran. So it's just like a carbohydrate where you would put a fluor and then have 10 carbons, and then put another fluor and have 10 carbons, that kind of thing. And it was just like a total failure. It was like almost like a ladder, but it rotates too much, it non-specifically binds, it quenches, it does all these bad things. So nature knew what it was doing because it had that kind of isolation chamber for each fluor.
Yeah.
But if we could synthetically not have to still derive this stuff from bacteria, you know, they've tried to make it in eggs, to do protein expression in eggs and things like that, trying to get it to have a good folding, but it's not as good. I never encourage people to do their own conjugation with PE. It is just that heterobifunctional cross-linking to the hinge region, but it needs to be cross-linked. The likelihood that your antibody is going to survive as long as it would if a manufacturer made it for you, you might get it to work right away, but I don't think your shelf life, I'd have to do research into it, but it's never one. I think anybody can conjugate, you know, their antibodies to fluors. Like that's 2 hours of a conjugation kit to conjugate Alexa 647, for example.
Right.
But tandems, especially with proteins, that's too much, you know, like, and they have to understand that as we get into spectral and we have 45, you know, Phytonics just did 45, you have to have every rando small target, every small marker conjugated to every tandem and every, you know, every polymer. You need to have that grander selection to pull from. So the nice thing about PE though, for people doing like extracellular vesicles and like photon counting and stuff like that, is that you will always have one PE to one antibody. You'll never have two antibodies to a PE, and you could never have two PEs to an antibody. It would completely lose function. So, you know, as long as you can purify that fraction, you know you have a 1-to-1, versus using the rest of the probes quantitatively. I definitely have issue with that. You know, you do not know that you have 4 Alexa Fluor 488s on an antibody. You have a range of 2 to 8. You know, so to say that you could say in a larger sense in this pixel, I'm quantitating information on an average and I'm qualitatively comparing it to others, but it's way different than having a 1-to-1 conjugation with the antibody. So another interesting one that's coming, you know, like it's been around for a while. I don't know if they'll ever make it to like a commercialization phase, but it's using chlorophyll or other kinds of organic fluorescents that can be extracted from bacteria. Chlorophyll is unique because it is so far in the near infrared, which actually Ryan's finger is blocking. It's like around the 625, 650 range on that spectra that I'm showing there. And so it's definitely a really unique structure. If they could ever make it more soluble, I could see them using that for a polymerizer.
So quick question. So you mentioned that people try to use eggs to generate their PE instead of bacteria. And maybe I missed it, but like, what's the downside of using bacteria? I just thought.
Oh, sorry. It just doesn't fold. So even people using baculovirus, where that should help with more complex proteins and better folding, they've never had good success with using an artificial protein replication method or protein manufacturing method.
So you kind of have the right protein. It's just not the right configuration. It doesn't do what it's supposed to.
Well, yeah, because it's just, each of these is one, okay, so that structure of PE, I want to say it's 5. Oh yeah, it's a hexamer, it says on the top. Each of those leaflets is assembled into that structure. So you have to first have the leaflet assembled correctly before it then combines.
Okay.
Way hard, right? Like, and then you have to cross-link it in order to get it to be like stable for any period of time. So yeah, there's a lot of limitations with proteins for, the kind of the uses that we would have for them. Okay, go to the next one.
All right, tandem dyes.
Yeah, I'm not going to spend a lot of time on tandems. Like all of this stuff is kind of old news, but that's the leaflet that I was telling you about. Like that's just one segment of that structure. But the most important thing as we talk about the DNA backbones and brilliant violets and any other sort of advanced tandem that we're ever going to have in our hands, everybody has to understand collisional quenching. It is the thing that most limits everything that we do in reagent development. And that is simply that you can't have molecules, you can't have two molecules that resonate energy. You can't have them too close to one another, or a photon from one cloud will collide with the cloud of another, as is normal. But if they're farther away, the likelihood of that happening is less. So the idea is either like PE to have a protein matrix around it so it's protected, or to have them far enough away from each other. But distance is a problem for tandems. We're talking about angstrom distances for frets. We're talking 10 angstroms, in order to have them so close together that we can have an efficient transfer of energy. But the more we do that, the more fluors we'll need to get every single one of those phycobilins in there. Every single one of those, we want a fluor next to it. And that's not what happens. Even on PE, you're going to have a very limited number of acceptors. And so each of those donors is all fretting energy to that single acceptor. When that single acceptor photobleaches or becomes oxidized, they have nowhere to go. They have no one else to give their energy to, so they're just going to spill back into their original emission, wavelength. So that fretting distance is so important in PE and all these proteins. We can't control how close we get to that chromophore, right? It's totally random. You're just trying to kind of loosen up the PE a little bit, jam down the acceptors into that structure, cross-link them, fold it back up, and conjugate it to an antibody and hope for the best, right? Versus if we could engineer a polymer so that we knew exactly where our fluors were placed, we knew exactly the distance.
So
that would be so much better.
The tandem dyes in spectral flow are kind of been super annoying because like from one experiment to the next, like everything's gonna be different.
Yep.
But that
yeah, it still amazes me that people talk about banking their controls and using them for later experiments, because especially when you get into the near infrared and those fluors become more sensitive to oxidation, your tandem degradation, is going to be entirely dependent on you and what you did.
Is that something that's driving the industry at this point? Like some getting rid of those tandems and just try to go at it, a single molecule thing?
I mean, I'm sure, Photonics or some of these new reagent manufacturers, I'm sure that's what they would love to do. Who wouldn't love to replace all of the business of protein? I mean, especially, I always say Percy-Psi 5.5 is the easiest target because it's such a labile molecule. If we can replace that, we can replace PE-Psi7, replace APC-Psi7. There's such low-hanging fruit. But the likelihood that we're ever going to get to 45 with only one company's reagents is super low, right? Like you can't have all the non-specific binding characteristics of the brilliant dyes in one assay. They will just be one glom of, like each of these different kinds of chemistries, they have their own fallibilities, they have their own problems. So if we only have one kind, we have an amplification of only that one problem.
Right.
So it's better I think, to continue to have a diversity. We also don't need them all to be bright. We just need them to be clean. which is kind of ironic. I don't know which one is next. Okay, wait. So, so with this one, what I wanted to point out was, I don't know if you've ever heard of Biodium.
No.
They are a great company. They're like Molecular Probes used to be in the very beginning. They've got some really great chemistry. They have some dyes that are pegylated already. That peg is just like the protein. It's protecting the fluor to keep it separate from other fluors so that you can get a higher degree of labeling. Not all their fluors are. A lot of them are just Me Too's, like the Alexa fluors. But I think this one is CF640R and 633. You can see how in this graph right here, the photo bleaching seems to not be so dramatic. And that very much is a reflection of the degree of labeling. Like when you have more of something and you talk about the normalized, see on the left-hand side, it says normalized fluorescence intensity. It would be different if it was not normalized, if they were actually taking relative intensity units and comparing them to each other. And that's kind of what I mean. When you have fluors that you can get them closer to each other to fret, so therefore you can keep them farther away from each other. When you can get all of those things together in the same package, then you can have the brightest thing that also then seems to photo bleach the slowest. Does that make sense? So that's the ultimate sort of package that we're looking for, but it's very much dependent on the chemistry of these synthetic fluors.
Right.
Okay, so we'll go to the next one. All of that's just fundamental for talking about the other backbones. All right, so nanocrystals, I still think that there's a beautiful future in nanocrystals. These were always at the whim of, I always use the example of eventually our iPads will be flexible. You know, they're not gonna be hardcore metal semiconductors on a plate. They're gonna be probably graphene or something that can be rolled and manipulated, all within a sheath of protection from oxygen. That's going to be the biggest thing with all photostability is if you don't have a source of oxidation, then you can have, violet type dyes, or LEDs, which is what brilliant violets are, OLEDs. You can have them inside that encasement so that you can roll it, and it's just going to be way cooler. But right now we're still dependent on all this metal to conduct that energy. All right, so QDOTS has a ton of fallibility. The size of the core isn't so big. It's all of the surface coating, the amphiphilic biopolymer that allows it to be conjugated to antibodies. And that's the biggest problem for all of them. I mean, even if you had a silica particle and you could get it aminated to the surface so that you could directly conjugate to the metallic core. Do you understand what I'm saying? Yep. Have you ever seen those in other applications? Because actually most of this isn't for biology. We're just beholden to them for whatever comes out of electronics and personal electronics industry. But that doesn't help us because that's going to drop straight out of solution. Just because you give me a handle that I can conjugate an antibody to doesn't mean that it's not going to just stick to the first hydrophobic compartment that it finds, or literally just drop out of solution in a big mess at the bottom of your tube. But what I think is cool is they're taking the concept of the nanoparticle, and I don't know, Ryan, does AbbVie do anything like this where you're using nanocrystals or nanoparticles to deliver chemotherapeutics or oxidative stress so that you could ablate a tumor? Like you're making them into carriers, not just, and they could be fluorescent, but they could also be magnetic for, what would that be, MRI? For different kinds of imaging in humans. And with those, lanthanide doping is really becoming a thing. You can have these different combinations of lanthanides that will transfer energy to one another in the core of the nanocrystal. But our issue, they can do whatever they want for electronics with these cores. It's going to be super cool and it's going to help us. But unless we can get them small enough, so smaller than like, 10 nanometer of the entire probe, not just the core, we need the entire slathered with, biopolymer kind of stuff. We need that to be 10 nanometers or under 20, at the worst case. But the lanthanide doped ones, the graphic that I'm showing you is kind of the process by which it hits, it's fretting in a different way to take the energy from one excitation state through. Did you guys hear, what's the dude's name? He gave a great Cyto, the virtual Cyto this year. He was in the Asia Pacific, but he was talking about upconverting nanoparticles. And you would think that that shouldn't be possible because the energy that's going in is actually going to be less than the energy coming out. Are you familiar with multiphoton microscopy?
No.
Okay. Multiphoton microscopy, you have a pulsed laser that is focused on a particular spot in space, in 3D. And you're hoping for a certain possibility that two photons of a very low energy state, like 900 nanometers, would collide at the same time to give you the energy equivalent of 450 nanometers. Does that make sense? Because the higher the number, the lower the energy.
Yeah.
So when they collide together, you're hoping that you're going to combine their energy to like 450. Mm-hmm. So that's how we do deep tissue imaging, multiphoton microscopy. Upconverting nanocrystals are kind of the same thing. It's just that because they're semiconductors, they're absorbing that 980 nanometer wavelength energy. And within the nanocrystal itself, they're just generating until eventually they come to, based on that lanthanide, each of them have their own spectral characteristics. Then when they reach a certain point, then they'll emit a photon at the designated wavelength for that lanthanide. So that's what upconverting nanocrystals are. It's not exactly the co-localization of two photons at the same time, like multiphoton, but it's a similar concept. You're using that much lower wavelength, higher penetrating, excitation source until eventually you can motor up enough energy for the upconverting nanocrystal to emit. So I think that those are super cool, but again, particles, conjugation.
So how low of excitation would be best for that?
I think they're doing between like 800 and 950, or maybe even up to 980, which is funny. That's low. Yeah.
But would it be better with lower? Or does that defeat the purpose?
I think that would kind of defeat the purpose. That's a really good question. It's not in my wheelhouse to know why they make one decision or the other. This is like pretty hardcore nanoparticle materials engineering stuff. But I would assume that it's similar to why we only use 3 lasers. We can add more lasers, but all we're going to do is cross-excite the fluors that we already have. Do you remember in Boston, they had an entire seminar series on compensation, doing flow cytometry with no compensation? Because it was people that had enough money to have machines where they would have 5, 6, 7, 8 lasers, and then they would use all the same dyes for microscopy because they could discretely excite them. That's kind of what that reminds me of. Compensation. All right, so the last one there. Oh, well, there's actually 2 kind of. Nope. What? Go back. Go back. Yep. Carbon and graphene particles are pretty fun because you can also dope lanthanide into those. You're just creating this matrix because that's what everybody's trying to do. How can we polymerize? What kind of things can we use to create matrices, whether they're sheets or tubes or strings or whatever they are? How can we harness them so that we can then dope them with the individual fluors with the signature we want spectrally, right? So those are pretty cool. You could have like literally a silica dot, which would just probably follow a solution. And then surface coat it, but it would be better to kind of have them, the graphene sheets sort of, and insert the copper or the manganese or the ytterbium or terbium or whatever the hell, put them all in there. And also because you're fabricating it, you have possibility of knowing where your fluorophores are. Not quite to the level that the DNA backbone does, but you can have a little bit more precision in knowing the placement of each of your fluorescent. Okay, and then of course, you can make tandems out of them. So all of these things, they're all related to each other. They're all just these little, as their research grows, they're like, hey, that's a good idea. I bet you I could do that with my silica particles or my iron particles or XYZ. So I still think there's a lot of potential here.
We're already talking about how big these things get. When you turn them into tandems, does that like make them balloon up?
Actually, no. The organic fluorophores are about 1 nanometer big.
Mm-hmm.
So your acceptor dyes are not doing much to change the size of the overall fluorescent compound. For the next one.
Brilliant.
Yeah. All of this, since I helped develop this fluor, all of this is from the patent. All of this is publicly available. I'm not saying anything wrong here. So like an OLED, an organic light emitting diode, that's what these are. They're semiconductive circuits it's just that they're organic conducting circuits. So the individual subunit is here, the optical segment. And then the question is, how many of those segments do I have in a single strand? And what spacing distance do I need between each of those optical units before they start to either quench each other or homofret with each other? Do you guys remember what homofret is?
I don't. Ryan does. Ryan's muted. Ryan said,
with yourself.
You fret with yourself. Well, there you go. Gosh, you're so smart. Yeah, you fret with yourself. You see this a lot with blue dyes, like UXT and DAPI is known for homofretting. And we don't want them to change spectral characteristics just because the distance between two fluors. So they just need to be individually existing with their own discrete properties, in a big, long polymer chain. So that's kind of what the first part of this graphic that you guys have probably seen before. Sarah was using this. Each of those optical units is an absorptive unit, and then you can attach an acceptor dye. And you can also somewhat know where that acceptor dye is, but we're not going to get into manufacturing on those. But it really, that last year at Probes, all I did was Q-dot conjugations. I swear to freaking God. It was so painful. Like when I started at Molecular Probe, at BioLegend, and they're like, hey, we need to support 18 color flow. And I'm like, well, good for you. I don't want to do Q-dot conjugations anymore. Like, this is not a way for this industry to go forward. And then within 2 months of me working at BioLegend, Sirjan contacted us, and they were on their last leg. And he even got a speeding ticket on his way to deliver his first seminar to us on the chemistry. And it was just like somebody dropped a bomb. Like, it was still one of the most amazing feelings I think I will ever have, is sitting with just one other person waiting for this dude to show up, being like, seriously? Like, doesn't he know our time is valuable? And then listening to him talk about these monster molecules and all the physical chemistry that he had on them. And I'm like, holy God, like, we need these now. I walked into my CEO's office and I'm like, if we don't have this, first of all, we're not getting Q-dots. Like, this is going to change everything. And it did. And so then everything, I think, is very much under pressure from, the intellectual property around this is just too tight. Nobody can make this better without BD loosening control. So still so much work to be done on these, like, so much fun. But the important thing is that, again, blue fluorophores are small, so the polymer can be longer. If I'm going to make a brilliant near-infrared polymer backbone, it's going to be like a square. It's going to be stout, wide. There are limitations as you're going to be changing the physical characteristics of the polymer by having the backbone contain much larger fluors that would allow you to be in near infrared. So you can always transfer that energy. Having them as acceptors is not a problem because they're not integral to the backbone. Does that make sense? So when, we've always seen them talk about stuff at CyTO under the emerging technologies banner. Mm-hmm. And the, brilliant yellow green that we've been waiting on forever. Have they ever released that?
I don't know.
I don't think officially.
I can only imagine what that looks like. I mean, brilliant violet is like a horseshoe because of an internal kink to the structure. And I've always said this in my talks that, when you get to anything that's going to be longer emitting, it's going to become like a tighter Cheeto, like you're not going to have as many units. So it's not going to be as bright. Simply, you can't make them as big or as bright as you can in the smaller dies. So, but there, a lot of the issues that always frustrated me trying to make these into something more useful for all of us is that they can't be lyophilized together. They just, I was seeing these uber brights that made the threshold on my cytometer correct. What did you call that? Threshold. Where your threshold is reset because the cytometer detects more background than it was expecting to see in QC?
Baseline restoration.
Yeah, baseline restoration. That's what it is. Baseline restoration. I was seeing that all the time, and I'm like, how can my unstain be brighter than my stain sample? And then Howard Shapiro wrote to me and was like, yep, you're seeing baseline restoration. I'm like, oh, that sucks. We can't lyophilize brilliant violets. And then, the nonspecific binding using any sort of buffer during staining, that's not helpful for any of us, right? It'd be better to fix the problem than have to use a buffer. And then, size is always an issue just for intracellular and internuclear type detection. So the P dots, which is probably what we were talking about, that's probably may or may not be what Bio-Rad has. I have no idea. But I believe that they have the IP from Daniel ChUG on WashU. So basically imagine that you had a strand of the brilliant violet, but that you configured it so that you could make it lap onto itself. So what's the right way to visualize that? It's a polymer dot, right? you instead of, you're not coiling it or anything like that. It's not as regular of a formed dot as that would be. It's more like they're all just layered on each other, but it reduces the overall size and space while keeping a certain high level of brightness. Solubility can be an issue, but
it is a linear polymer that is coiled up somehow.
Exactly. Yeah, kind of always thought as like a big piece of snot. And then you have to react that in order for it to be able to be cross-linked. In this case, they're doing it with an EDAC reaction. So I think that also probably has a lot of potential in the future. I don't know if that's exactly what Bio-Rad has, but it would be a place for a lot of development to come as well. So go to the next one.
Well, can you clarify something for me before we move to the last guy? So whenever we talk about UV dyes, some of them are single molecules, some of them are tandems. And so if I understand, and maybe I don't, that's likely. The molecule here, the bunch of your string that will accept the energy from a laser and then transmit to a dye, and that's your basic molecule BV 421 or 510. But then if you have a tandem, you just stick something and then you on that thing, and you stick another one of these dyes we talked about before, and then like a site 3, site 7, site whatever.
Yeah.
And that's the one to accept the energy. Okay.
Exactly. Like, and that's exactly what I was talking about before with the synthetic organic fluors. Mm-hmm. The optical unit is a simple organic fluor, right? Of some sort. It's just polymerized into this backbone. And then you have all these other elements, like the little top part there, that are helping give it space and solubility and conjugation ability, right? And then the acceptor on the outside, also just a synthetic organic fluor. We're always tuning based on how we can structurally modify those cyanine, xanthine, coumarin derived fluors. So we're always limited by our own visible spectrum, you know?
Right. Okay. Cool.
But that's also, sorry, one other thing, because we get this a lot now that people are starting to get, you know, with the photodiodes going up to 1000 nanometers, getting dyes above 750. Like, have you guys ever used IR 800 or any of those?
Yeah.
Yeah, they're super, super big, super difficult to keep soluble, super difficult to have them resist collisional quenching because they're so big. Like, to work with them, because I did APC Fire 810, that was the farthest red we had ever gotten, and that's because we found an acceptor that defied expectation. We also failed probably 4 candidates, you know. It's very difficult to continue to create fluors in the range that will max out technology in that application.
Oh.
That's always kind of what bugs me a little. For live dead, it'll be fine. I could take an 800 emitting dye, conjugate hundreds of them to my dead cell, and get it to show up on a near-infrared laser.
Mm.
But when I'm conjugating those as an acceptor to a donor, I can't get enough of them. Does that make sense?
Yes. Quite. Right.
However, as an upconverting nanocrystal, those lasers might be very interesting. Just saying. Mm-hmm. All right, so this is where a lot of the technology is at. This is nature's second polymerizer. Well, actually, it's probably the first one, right? Because it's DNA. But, so you can have, most of the work that you'll see is double-stranded because it holds everything in a particular rigid alignment. So this graphic that I'm showing here on the left, that you see how it's saying stacking of those amino acids? No, of those nucleic acids. Those are what we're gonna mutate. So those two, what's the right word for that, you guys? Is it the nucleoside?
Nucleus what?
Because you have the phosphate backbone and you have the nucleoside, right?
Ah.
Or is the nucleoside for RNA? I forget.
I don't know. Somebody watching this will be screaming at their screen right about now.
Oh my God, they will be for like all my poor use of chemistry. They're gonna be like, coumarin's not a family. It is to biologists. Okay, so so you have your phosphate backbone intact. You're actually just modifying the nucleotide. You're going to substitute fluorophores at a particular sequence, but just like when we were able to make fluorescent modified DNA and RNA back when we were at the beginning time of sequencing and the genome, we know exactly how many nucleotides we'll have before we have one that has a fluorophore. We have ultimate control to tune and know exactly where all of our fluors are. And we don't just even have to insert fluors into those nucleotide spaces. We could have some other kind of derivation, like we could have an amine or something else that gives us a tag or a hold to modify something later. It is like the epitome of controlled synthesis of polymers. This has been going on for all of the 2000s. Like, we are just getting on this. Did you guys ever talk to the phytonics when they first came to Cyto? And they were like, look at this flow cytometry stuff. And we're like, whoa, fluorescent DNA. Where both seem so obvious to themselves, but not to each other. So Eric Kuhl is probably one of the funnest chemists in this area to read his papers. He was actually using these fluorescent nucleobases in order to sense RNA and DNA replication, damage, that sort of stuff. That was his research, his biological research area. But at the same time, he was also like, if you see him give a talk, he'll show you the spectra of integrating like 6 different spectrally discrete subunits in a single strand of DNA. So he would have these like little peaks as if this one biomolecule, do you see what I mean? Like, it was so cool. But for us, that would suck. Although it would give us this really unique fingerprint. We don't want something that excites off of every laser. We want it just to excite off one laser and emit in one spot for how unmixing will work. Mm-hmm. But lots of other applications like unmixing and microscopy, that could benefit from this. You could make any combination you wanted. It's just so cool. And the phosphate backbone, it keeps everything soluble. So as something gets bigger and we have trouble keeping it soluble, that's not a problem here. If anything, you've got to maybe start worrying about having too many phosphates and starting to like make an acidic solution where you might want to cap some of them or something like that. I don't know if any of that's an issue yet. That would be, somebody would have to do that work. I don't know who. So does that make sense to you guys? So you create, you know, in this case with Eric Kuhl, he's just creating single DNA strands. The other reference that I have to the left of that one is people determining the distance between each of those base insertions. What's the optimal distance to create fret? So if you wanted to use these as sensing probes, you might have something like a sequence that would be the cleaved portion of that DNA in between Psi3 and Psi5, for example. But for us, we just want them as reporters, right? But that's the kind of research that was going on before now. And then imagine just like with just like with brilliant violets and P dots, you can then hybridize oligos to each other. So I think the phyton is like B. I think they have that on their website. But you know, as long as you know exactly what your sequence is, you can say, oh, on the second sequence, here's the hybridization rate, you know, zone. And then maybe, you know, you can create literally any structure you want. If you do a Google search on DNA origami, you have people literally DNA printing boxes, smiley faces, like, because they're just having fun. They're doing proof of concept to say that you could create any 3D structure just by knowing how they hybridize together. So that's super fun.
So has somebody
linear.
Has somebody figured out kind of what the optimal 3D structure is to keep things, the dyes, you know, close enough but far enough away so there's not quenching, etc.? It seems like that could have been, you know, step number one.
Yeah.
Right.
So I mean, I don't know everybody that's doing everything here, right? We only know people that like phytonics that knew to approach biomedical research with their ideas. Well, okay, trying to think of, all of this is in the literature, I think. So maybe we should stay away from that. But okay, so say that you're gonna have 2 sequences that hybridize. Okay, so like B, you've got a phyton. Each of those strands might be, one is a donor strand, one is an acceptor strand. You need to be able to manufacture those in high throughput. So your assembly and knowing that distance, what's the right way to describe that? It has to be practical and not just the best confirmation, right? How do we're not just making something that's special once. It has to be something that we know that we can self-assemble, and it will always be without fail with its partner.
Yeah, I mean,
probably not the best one to talk about this with either. I would, you know, I'm sure like the dudes at Phytonics would be.
Yes, certainly production has to be part of the R&D process, right? If you can't produce it reliably and quick enough and in high enough volumes, then you know, it's basically just a research paper exercise, and it's not a product then.
Yeah. Well, and you also want to assemble it into, you have, if there's one phyton, those are still 4 linear segments of oligos. Do you know what I mean?
Yeah, but what if like taking that phyton and then putting another one that's in the 3rd dimension, right? Does that like double your amount of fluors that you can put on together?
Definitely. And also but it's also a humongous structure. These are already going to be not the easiest things probably to get internuclear.
Yeah.
So how do you kind of have the highest degree of polymerization possible with the smallest distance between each of those optical units, just like the brilliant violets, and the assembly of them is still soluble, manufacturable, consistent, small.
Okay, so here's the $64,000 question for you. Of these various backbones, we have DNA origami or polymers or carbon fiber structures, whatever. Do you think there's going to be one that comes to the top and takes over, or is it, a mix of everything will be good?
I think that you're going to find the DNA. It's the easiest and least expensive thing to fabricate, probably. I don't know how much graphene costs to fabricate, but a lot of the graphene particles, you have to actually cut them and you have to shape them, that can't be very helpful. That has to be super manufacturing impeding. So I think the reason why we are seeing the 2 technologies that are using the DNA backbone, why they're the ones we see currently, is because it's that much easier. We know it's biologically compatible. It's not some materials chemistry that we're trying to apply.
Okay, so you said that the 2 technologies, so the phyton, what's the other one?
The Caravia.
Oh, Caravia. Okay. So do you know the difference between those 2?
Yeah, it's on the slide.
What was that it?
Yeah, so the Caravia, we call it Caravia. David call it Caravia.
I'm French Canadian, so
that's
true.
That's
probably the French pronunciation.
I have to say, I was working on this project for over 5 or 6 years, and we only got the name for it right before they commercially released it. So what does it mean, like peace or something?
It's a brilliant, no, it means sparkling in Japanese.
Oh yeah, sparkling like a star, right?
Now, quick question. So the sequence of the DNA will impact the structure, right?
Mm-hmm.
So I thought I understood that the general goal was to get that molecule to be used both in flow cytometry, say, and then you kind of throw that into an NX genomics type single cell sequencing thing.
Yes.
So that's not as easy as I make it in my mind, I assume.
Really? Why not?
I don't know. Everything is difficult in my mind.
For the fluorescent portion of it, it does not matter what the configuration of that DNA is.
It does not? Okay.
It only matters that your fluors are inserted at a certain regularity away from each other so that they're each optically independent.
Mm-hmm.
Outside of that, it does not matter what that sequence is. As long as it was in that graphic that I showed, they have to stack. So, but that's what the double-stranded DNA is doing, is creating a space for the fluors to be stacked like this. Versus normally when you have a resonation of energy, the fluors are on the same plane as one another when they're conjugated, and they're resonating energy across the plane. These guys are resonating energy up and down a column. It's so cool. I don't know if they can do it with RNA. And, honestly, those 2 things that I just presented, those are not exactly what either Phytonics or Sony's dyes are. Those are what's published in the literature. That's the backs upon which both those technologies stand. But I'm positive that they probably have synthetic DNA manufacturing. I don't know what all the ins and outs are, but all of them had published IP or, accepted IP that went above and beyond what was published in the lit. So, you know, and we were never privy to that at BioLegend of what Sony did to distinguish themselves from Eric Cool's stuff, for example. We just know how they're stacked and, you know, how they perform, that kind of stuff. But if we can use those, if we can use DNA backbones to get rid of the issues that we have with PE, APC, and phycocyanine, that's a big push for the future. But then we're going to start to be limited to overlap again. We need these to be discrete, but that's what we want. PE is not discrete, right? It was great when we had 3 lasers and all we cared about was getting 8 colors, you know, that's fine. But now it's too promiscuous. We need to have these backbones that are only excited by a UV and only excited by a violet and not by each other, you know? I mean, these might even displace the brilliant violet dyes for that reason, for that lack of promiscuity and excitation.
So I feel like I'm going backwards in my mind here. So help me come back to present day here. So the polymer dyes. So I know, you know, brilliant violet and whatever, let's say brilliant blue. The polymer backbones are different, obviously, because they absorb different wavelengths of light, correct? Or how would you say that?
I wouldn't say the backbones. I don't know for sure because I didn't work on brilliant blue. So this is just my hypothesis based on what I understand about the polymers. Just putting that out there. Mine's not the word of God on that one. BD is, or Brent Gaylord. The optical unit will be different. Absolutely, between each of those. That doesn't make the polymer backbone different, right? There's still a lot to encase and polymerize that optical unit. And those linkers were just as important as the conducting unit itself, because that was the same problem we had at probes. How do you get 2 fluors to stay isolated from each other is just as important.
So yeah, so I get the, right, so anyways, you're gonna have some portion of the brilliant whatever that dictates its absorption. And then you're gonna have some acceptor dye that's pretty much the same off of UV or violet or blue or yellow that gives you the fluorescence. Okay, so I understand how that can kind of segregate things across laser lines. So how does that happen on the DNA side of things?
So you have 2 different options with DNA to create the tandems. That's what you're talking about, is the tandems, right?
Yeah.
So you could have just a handle modification, like an amine on, you know, and you have a 3' and a 5', you have NH2s or carboxylates, right? So you always have 2 handles on either side, but you could incorporate more with modifications. So you could attach your acceptors to the outside of the strand of DNA, or what would make a million times more sense is to embed your acceptor within the structure of the DNA. You know, we think of it always as being a donor and the acceptor is like on the outside, but that isn't necessarily. It's a million times better if I know, I'm just making this up, this is not the distance between things, but I have my optical unit, I have a smaller linker size to another acceptor fluor, and then another limited size to another donor. Those 2 donors will both contribute to this acceptor. And then I've got a bigger link so that that's one optical unit together. That tandem and that fret is working. But if that one gets photo-bleached, then those 2 are now on their own. And they'll emit where they're supposed to. But that would make a lot more sense. And also it would protect the acceptors from oxidation. You know, they're gonna have an equal experience with their aqueous environment and their oxidative sources as the donor will. Versus right now, because we can't literally open up the PE and stick down the acceptor inside it, which would be really helpful, those acceptors are the first things to experience oxidation from the aqueous environment. So you're leaving them vulnerable So if they integrated, you have a more stable probe generally.
And what are in the context of the DNA setup, what are the donors?
Oh, I don't know. You mean like what is Photonics using for donors?
Yeah, or what would you use as a donor?
You could use anything.
Anything?
Oh my gosh. Literally anything, because your phosphate backbone is making it all soluble. So probes that maybe you had to stay away from previously because they were too hydrophobic, like Bodippies, you can use all of them now, because that's great because they're a lot smaller. So again, with the the size of the floor is going to determine how thick the polymer is. But okay, so the other thing that's really cool, say that you had a strand of DNA with only coumarin as the donor, something that has no 350 excitation and it doesn't have any 480 excitation. It's just, coumarin is 350. It's just 350, but not 405 or something like that. Something that's really selective for just one of the lasers. And you put Texas Red on it, or something that's like Alexa 594. Discrete excitation, discrete emission. With the siliconized rhodamines, you can cause redshifting that the spectral cytometer will be able to see. So this is what we should have done with the brilliant violets, if we could have, is discrete acceptor, discrete emitter. Now have a 488 polymer, shift that Texas Red just a little bit, like 5 to 10 nanometers, and you'll be fine. Because just that little bit of difference on a discrete donor, it will make all the difference in the world. The issue I think that we're seeing with spectra right now is having too many donors that are too promiscuous, and then the fluors have to be 20 to 25 nanometers apart for them to be usable. And they have to be bright, because with spectral, you're photon counting. Everybody's sharing a certain photon threshold. If your dye is too dim, it just isn't going to register next to a that has a higher output. The spark, what did we call it? Spark 15? You know, those ones that are real close to other more potent fluors. You know, sometimes in spectral, to have it on something so abundant, have enough photons of that spectral fingerprint to be able to accurately unmix them. This is fun. I love dyes.
We're approaching the end of the allocated time we gave ourselves to record this. So what would be a nice ribbon to wrap all this up? Like, would you say? Because we debated the title of this podcast, and I suggested Fluorophores. What are they good for? And you went with the future of.
Absolutely.
Would we say going forward we're provided with fluorophores that have a more narrow emission, specific excitation, and so overall it's going to be reducing the amount of noise that we're going to generate.
That's what better be. Better is going to be more discrete excitation. It's going to be a range of brightness that we can control to best match it to the, you know, like one of the things we talked about with the Kiravias, not that they're ours to do that work with, is, you know, even creating 2 different versions of these kinds of polymers, those for low abundant antigens and those for high abundant antigens, so that you're not overwhelming another partner and you're not less than another partner. It's that tunability. You know, I think that was a big catchphrase. It's becoming a big catchphrase over these last few years, is our ability to control and tune the features and characteristics of our probes to best match them to the biology. You know, your tools shouldn't be the thing that dictates your assay. You know, the biology, the question you want to ask, that should be the limiting factor, not, you know, not the solubility or the size of a tool.
Right. Meanwhile, we still have the same molecules we're going to have to play with, so we're kind of still some limitations yet to all this development.
Yeah, until the next instrumentation revolution, right? Mm-hmm. Like Raman scattering, like all the people using non-fluorescent approaches, you know, or single cell sequencing just takes over flow.
That's expensive.
Yeah.
All right. Well, I think we're going to wrap this up here. Kelly Lundsten, was delightful to have you here. Thank you so much for your time.
Yay, thanks guys.
I'll find you on cytometry.me. Again, kudos on the grabbing that. My brain going away. There you go. Thank you. All right, Brian, thanks to you again.
All right, thank you.
All right.
Bye.
Nice talking. See ya.